Now we are going to start with the next class. Hope the screen is visible. Yes, is it visible to everyone?
Yes.
Yes, sir.
Okay, fine. So students, we are starting with the next class of the anticancer agent, that is the organoplatinum complexes. So these organoplatinum complexes is further divided with the two drugs. Means there are two agents in this organoplatinum complexes. The first one is the cisplatin and the another one is the oxaliplatin. So there are only these two agents which we have to cover in our today's lecture. So these organoplatinum complexes, these are the antineoplastic agents, and if you check out now, this is the structure of the cisplatin. Okay, this is the structure of the cisplatin. So if you check out these agents, they contain an electron deficient metal atom. This it contain an electron deficient metal atom. In this case, it is a platinum. So this electron deficient metal atom, it act as a magnet for the electron rich DNA nucleophiles. So as we have seen in the nitrogen mustards, organoplatinum complexes, so they are the bifunctional, and these nitrogen mustard and organoplatinum, these nitrogen mustards, just like the nitrogen mustards are bifunctional, so just like nitrogen mustards, these organoplatinum complexes are also bifunctional, and also they accept electrons from two DNA nucleophiles. Okay, so just like these nitrogen mustards, they are also bifunctional and they can alkylate two DNA strands at a same time. So either intra-strand cross-linking takes place or the inter-strand cross-linking takes place. What is the meaning of intra-strand? That is, intra-strand means within a DNA strand. Means within a DNA strand, they alkylate the two DNA base pairs, and inter-strand means what? That is two different strands of the DNA, right? So these organoplatinum agents like the cisplatin and oxaliplatin, they contain a electron deficient metal atom like the platinum, and this electron deficient metal atom, it acts as a magnet for electron rich DNA nucleophile. Regarding the chemistry, all the currently marketed organoplatinum anti-cancer agents, they contain the platinum complex with square planar geometry, right? If you check out, it's a square planar geometry. So all the commercially available organoplatinum anti-cancer agents, they are having the square planar geometry and they contain the platinum as a metal atom. This platinum is inherently electron deficient, but the net charge on these organoplatinum complexes, it is zero. Why it is zero? As it is as the platinum is electron deficient, but still the net charge on these organoplatinum complexes is zero, because the contribution of the electrons by two of the four ligands bound to the parent structure. So over here, these chlorine atoms, they contribute their electrons with the platinum, and hence the net charge on these organoplatinum complexes is zero. So before reacting with the DNA, the electron donating ligands are displaced like the chlorine as they donate the electrons to the platinum. So before the reaction with the DNA, these electron donating ligands, they are displaced through nucleophilic attack by cellular water. Okay. So before reacting with DNA, It is shown in the next slide. Yeah, if you check out over here. So before reacting with DNA, these chlorides are displaced with the cellular water. So in this case, one chloride atom, it is get replaced by the water molecule. When the displaced ligands are chloride anion, for example the cisplatin, the chloride poor environment of the tumor cell facilitate the process, driving the generation of the active cytotoxic hydrated form. So this one, the active cytotoxic hydrated form is get generated. Okay, and these both the chloride atoms are get replaced by the water molecule. Then later on in the next step, this water molecule is get liberated as a living group, and later on whatever the N7 of the guanine, it is get attack on these platinum complexes as they are the strong electrophile, right? Now this complex which is formed, which is a strong electrophile, and this complex is the, it is not strong electrophile. This one is a strong electrophile because it carries two positive charge. It carries the two positive charge, as both the, why it carries the two positive charge, because both the chlorides are displaced by the water. Both the chlorides are displaced by the water, as the chlorides they donate the electrons. The chlorides they donate the electron and they make this complex neutral, that is zero. The net charge on this complex was zero, but now both the chlorides are displaced, so there is no any atom to donate the electrons towards the platinum, right? So it becomes highly electron deficient, and hence it become a there is a more positive charge, right? There is a more positive charge, so it is highly electrophilic species. Once the highly electrophilic species is formed, then the nucleophilic group, like the N7 of the guanine of the DNA, it attack on that electrophile, and thereby the DNA base pairs is get alkylated. Right? So this is what about the mechanism of action of this cisplatin. So in this case, it was shown this is the structure of the cisplatin, and this cisplatin molecule, it act on the single DNA strand and to alkylate these DNA bases. So it is shown in this figure. Once this cisplatin alkylate the DNA base pairs, then later on these processes are get inhibited. That is the DNA replication is get inhibited, DNA transcription is get inhibited, then it results in the cell cycle arrest. Then also DNA repair mechanism is also get interrupted, and later on the cell is get dead, right? So this is what about the mechanism of action of the cisplatin. This is what are the theoretical parts. So I'm not going to discuss all these theoretical parts, so you just check it out. This is how the cisplatin act on the DNA base pairs. It is shown in this slide. And again, this is all the theoretical part of this what I have explained this slide. So all the theoretical part of this slide, it is given in this slide and in this slide. So check it out later on. Okay, fine. So like nitrogen mustards, right? We have check out the drug chlorothamine in the nitrogen mustards. So the chlorothamine activation, it is get inactivated by the sodium thiosulfate. So like chlorothamine, the cisplatin activation or cisplatin is inactivated by the sodium thiosulfate. Okay. Now the next agent is the oxaliplatin. Now this oxaliplatin, uh, it also contain a platinum metal atom, and it loses oxalate dianion in vivo to form mono and dihydrated diamino cyclohexane platinum analogs. The trans dihydrated diaminocyclohexane structure serve as the carrier for the cytotoxic hydrated platinum and extend into the major groove of the DNA when the DNA platinum complex form. So I'm not going to explain each and every part. So this oxaliplatin, like cisplatin, it also form a complex with the DNA, just like the cisplatin. Means both the cisplatin and oxaliplatin, they have the similar action. So this is the structure of the oxaliplatin, and it over here it is shown that how the oxaliplatin bind to the DNA. How the oxaliplatin it bind to the DNA. Over here it is shown, right? That is oxaliplatin. This ring is get opened by the hydrolysis process, just like in the cisplatin we have checked out. That is in the cisplatin, the two chloride molecules are get liberated and the water molecule get attached to the platinum complex. So in this case, again by the hydrolysis, the oxaliplatin is get hydrolyzed into this complex, results in the opening of the ring. Then later on, by the removal of the dicarboxylate anion, and again by further hydrolysis, this platinum complex is get attached by the two water molecules. So it becomes highly electron deficient species. Once, and as we know that once the highly electron deficient species is formed, then it can easily attack on the N7 or the O6 of the guanine of the DNA base pairs, and thereby it can alkylate the DNA.